So now let's try to understand the coronavirus spike in general. So you've already seen the SARS coronavirus 2 spike and you've seen all its parts. And this video is not going to be, you're not going to get much out of it unless you know your single letter amino acid codes by heart. Like if I point to a K, you gotta say, oh, that's lysine, that's basic, at least if you know the properties, or if I'm circling a couple of L's, you're like, oh no, that's hydrophobic. Yeah. Something that wants to be away from water. All right, so these are the parts that we've seen on the SARS spike. We've got an S1 region, which actually goes up to the furin cleavage site, which is here. And then we've got an S2 region, which is everything after this. The S1 region is going to contain an N-terminal domain, which is the big gray thing, several glycosylation sites, which are little purple N's, and it's actually that asparagine N where we're going to attach the sugar. There's a signal peptide, and it's cut off by signal peptidase and recognized in a whole process. And then you've got your receptor binding domain. That's the thing that's actually going to make contact with the receptor. So this is the part that is going to be most different when you're looking at different coronaviruses that have different receptors. It's a part of the virus that can be customized through evolution to sort of shift the virus from one thing to another. Or rather, when random changes happen, then accidentally when this virus bumps into things, because that's all viruses do, they're sneezed out or thrown out at things and they bump into them and they either stick or don't. This is the part that's going to stick. Then you've got your, in S2 region, you've got your second cleavage site, which is in SARS-CoV-2. It is TMPRSS2 that does that cleavage mostly in the lung. And in the intestines, things like TMPRSS4 will also do some of that, and several other TMPRSS proteins. Cathepsin L also can do this, but it would have to get pulled into the endosome and acidified for cathepsin L to be activated. So it can get cleaved earlier if there's TMPRSS available, and it has to wait till later if it's going to use cathepsin. Either one works. There are viruses that do both, yeah, or one or the other. Then you've got your fusion peptide. That's the little hook that's going in the membrane. You've got your heptide repeat 1 and 2. Those are the orange bits. And you got the region in between there where your sensor may be. You've got your fusion related. It's a CARC motif. And you've got your transmembrane. And then you've got your palmitylation sites. And you'll put a variable number of palmitins on these, or palmitic acids if you like, on these cysteines. You see the pattern here. They're spaced out just right so that they would all be on the same side of an alpha helix, so that they can all be facing up into the membrane together, and so that palmitoylating one doesn't prevent you from palmitoylating another. So there you go. It's a nice little anchor region. All right, so that's SARS. So let's look for these things in other coronaviruses. So if we're going to do that, the first question's got to be, well, do other coronaviruses even have a spike? That's a good question. So here are all the coronaviruses. We've simplified it down so there are only a couple of the coronaviruses you would know, the orthocoronavirinae. These are the ones that contain viruses like SARS-CoV-2. And the spike is going to be the yellow box, and you can see, yes, there's at least one yellow box in every single one. There are also other things that are basically spike-like. So you will get in some viruses a hemagglutinin esterase. It's not shown here, but that is a spike-like protein in that it will attach to a cell. It doesn't have the fusion part. It is only an extra attachment protein. There's also a class, and this is quite new, of proteins, these little yellow stripy guys right here. They have the attachment part, like the S1 part, and a transmembrane, but they're missing the fusion part, which is S2. So it's like a shortened version of the spike that would be only for attachment, presumably. And you've got at least one virus that has two, apparently fully fusion competent spikes in there. Maybe this allows it to enter two different types of cells that it needs to complete its replication cycle. Who knows? Yeah. Hard to say. But yeah. So if it's a coronavirus, yeah, it does appear to have a spike. So that's good, right? All right. So with that in mind, go see our multiple sequence alignments. And so this is a program that it would be very difficult to get installed on the class computer, so I'm showing it on mine right now so you can see it. What we have here is an alignment of, let's say, all the spikes. Now there are actually different groups of spikes. They're all related to each other, but they're far enough apart that you wouldn't like to say they definitely, work the same. You couldn't assume that. And the part that you won't see here is S1, because you can't align S1 for all of coronaviruses or even major groups of coronaviruses. Can't be done. It is fully customizable, and there is enough change there that it would be a fool's mission to try to actually align those. So S1 changes a lot every time there's a coronavirus, so much so that you can't align them. Now, what we've got here is an alignment program. It's called JALView. Here, we'll pull it down. See, oh no, it's a little too tall. We'll push it up. Nope, won't let me do that either. All right, well, it's JALView. You can see the bottoms of the letters poking down. And in JALView, we're putting our multiple sequence alignment with coloring that's going to pick out the type of amino acid. So cysteines, if they're always cysteines, that's our C here, are always going to be this sort of salmony pink color. And hydrophobics are always going to be light blue like this. Big aromatics are going to be this sort of, I don't know, it's like a muted teal color, like this Y's here. Polar amino acids are going to be green. Proline's yellow, glycine's orange, because they're going to be flexible regions or a bend in the case of proline. And if we ever get to acidic regions and basic regions, here we go. Acidic, or rather basic amino acids like K and R are going to show up as red, and acidic amino acids are going to show up as purple, and you don't see any of them here, which just means there are none conserved. There are acidic amino acids, but the way this coloring scheme works, there's a certain threshold. Unless you are conserved enough, it's not going to show you the color. And that's just a good thing for us, because it's telling us, is this actually meaningful? Because if you had the colors for everything, you'd say, wow, there's colors everywhere. That's really neat. Yeah. And I understand nothing. What you're seeing here are colors where things matter, where things are the same pretty consistently, or consistently enough. All right. So, in S2, what is not conserved is the cleavage site, the furin cleavage site, because only about half of coronaviruses have that. So that's not a good thing to align on. And some coronaviruses actually don't have a furin cleavage site or any cleavage site at the S2 boundary. They would only cleave at the S2 prime, which is the TMPRSS2 cleavage in SARS-CoV-2. What you do have in S2, though, in the top of S2, are a series of disulfide linked cysteines. And so you see absolutely conserved C column, C, 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 and look at that, there's 2 pairs of them. So these guys are going to pair up, and I don't remember which one pairs with which. You'd have to see it in 3 dimensions for it to make sense. But yeah, we've got conserved cysteines participating in disulfide bonds up there in the spike. Great. All right, and then we scroll down here, and this is the group that includes SARS-CoV-2, which is that one right there. And so you can see it's fairly normal compared to its peers in here. There we go. Now the next part we want to get to is the actual TMPRSS2 cleavage site, which is this, sometimes it's polybasic, sometimes it just has a couple basic residues like the K and the R here. Now you see that those are present in SARS-CoV-2, which is that one. There we go. And that is going to be the TMPRSS2 cleavage site. And I forget if it's between the R and the S or after the S. You can look that up. So we know that right after the cleavage site, we're going to have the fusion peptide. Now the fusion peptide is an amphipathic helix. An amphipathic helix is easy to recognize in an alignment Because you're going to have a pattern of conserved hydrophobic residues. And it doesn't matter which hydrophobic residue, but at that particular position, there's going to be a hydrophobic residue. And they're going to be conserved. It's basically every fourth or third apart. So if you count 1, 2, 3, 4, 5, 6, 7, 1, 2, 3, 4. Oh, it's trying, and it's actually shifted off by 1, so this is tilted a little bit. 5, 6, 7, and if you go backwards a little further, you can actually pick up another one. That is an amphipathic helix. You got one side that is hydrophobic, and the rest of it is whatever. That's why you don't see any color in the rest of it, or you see little scatters where like G is always colored, so just ignore the yellow and the orange in this case. But yeah, there you go. Amphipathic helix, that is the fusion peptide, and so the fusion peptide itself is conserved, but the TMPRSS2 cleavage is not. Now the fusion peptide we think probably doesn't work unless there is some kind of cleavage, and you do see a range of some basic amino acids here. There are proteases that like to cleave after basic amino acids, but there's lots of different ones, and these are all in different animals. And so, like, white bream fish, we may not know exactly what proteases that sort of thing has in its gills, which is where this would infect. And so it's unknown, but something is probably doing the same job. That's a thing that you can be fairly certain of. So yeah, TMPRSS2 cleavage itself not conserved. Cleavage at that site probably conserved. Fusion peptide conserved. Yeah. Scrolling on. After the fusion peptide, what's the next thing that you remember? Well, there's a lot of sort of spammy nonsense here, and this is just an area where it's probably going to be out toward the surface where the immune system may attack. You could have glycosylation sites there. Who knows? Yeah. A little hydrophobic cluster here. It's all these blues lumped up together. Great. And then we're going down, and then, oh, what is this? What is this? What we've got here is, do you recognize it? Little blue bars. Blue bars, like a little blue jail, and they're spaced out. There's a gap of 3 and a gap of 2, and a gap of 3, then it'd be a gap of 2. And sort of 3, 2, 3, 2, well, 3, 3, 2, 2, yeah. They tried. Viruses try. But basically, what you got there is a great big amphipathic alpha helix. And that is heptad repeat number 1. And it's huge. Look how big it is. Boom. And so it goes from roughly there to there, and it is conserved. What's conserved about it? The amphipathic side of it. So the fact that it is nonpolar on one side, which is what you're seeing in these little blue columns. And it's kind of built in 2 parts, and there's sort of a flexible region here. This gets bent over in some structures. So that it starts out like this, and then it's going to go like that when the fusion process is happening, when S2 extends. All right, so great, another marker. Now down in this area, we have found, look at this, we've got cystains. So that's another pair of cystains, and another cystine and a cystine. So we're probably going to have more disulfide bonds down here. And that's fine. This is just a little area where we're going to pinch these together and disulfide bond it, keep everything where it belongs. Look what else you have some of here. You have some of these aromatic residues, and you have some conserved, sort of conserved, histidines. There's not a particular spot where you can say, there's always a histidine here, this is the one that triggers it, because frankly, the virus doesn't need to be that careful. If there's a histidine in any number of places in this region and it gets protonated, that is going to be enough to spring the S2 so that we can start the fusion process. So this would be the area where you would look for those. Then you can see a couple of them that are showing up a little bit, and there are some everywhere, but there isn't a particular exact position where you always find them. Okay, let's get to the end of this little pinched off region, and we've got to slide this over. There we go, because we got to see our last major feature. Here's the last feature. You see it, you recognize it. That looks like an amphipathic helix. What's the amphipathic helix toward the end of the spike? Why, it's heptad repeat number 2, of course. And that's what this is. So you can see heptad repeat 2 is kind of small compared to heptad repeat 1, which is, yeah, this whole chunk from here to here. Yeah. And so this is going to bind to part of heptad repeat 1, and heptad repeat 1 is mostly going to bind to the other heptad repeat 1, so they're going to make the inside of the 6 helix bundle. Heptad repeat 2s are going to sit on the outside of the 6 helix bundle, about like that, and there you go. That's your spike conserved region. Now, there are other things that we should find after this. We should find the cholesterol or lipid binding region, but we don't see that. And that's because it's not universally conserved in coronaviruses. Or rather, they may be doing the same thing, but they may be doing it in a different way. It may be that there are enough outs, like enough ways to do it, that you don't have to do it a particular way. Evolution has been exploring this region for a long time, and they all work well enough to get the virus into whatever kind of fish or mammal or bird cell it's trying to get into. And yeah, there's probably, the bar is probably at different heights for different cells, and yeah, these things all work, so they're all real. After that, you would expect to have the transmembrane region. That is conserved. There's always a transmembrane region, but it's always just some hydrophobic amino acids, so it would look blue. The thing is, you can't really align which hydrophobic amino acid matches which other one, because they're all just hydrophobic. They change to other hydrophobics all the time, and there is no consequence whatsoever. The virus is fine. Just after that, you would expect to find the palmitylation sites, those cysteines, and you can find it in maybe half of coronaviruses. Maybe I'm being generous there. Maybe it's only a third of coronaviruses. The rest don't seem to do it that way. So do they have a different acylation modification at the tail? Hard to say. It could be. Or are they, bolstered enough in some other way that they don't need that extra acylation? Also a possibility. All right. You did your first coronavirus spike. Let's look at a second one real quickly, a second group of spikes, and see if you can spot all the things on your own. So we'll pull this one up. We'll slide all the way over to the beginning. All right. So do you remember our beginning part? What did we have there? 1, 2, 3, 4. Disulfides. Yeah, it's like Dora the Explorer. Yeah, let's go. All right. Then we've got two things, both of which could be the fusion peptide. If this is the fusion peptide, it doesn't really look like the regular coronavirus fusion peptide. It's like a whole bunch of hydrophobic things in a row, along with some small amino acids. Glycine is a very small one. It has just a hydrogen for a side chain. That is what you see in influenza A virus, hemagglutinin. Its fusion peptide is not particularly hydrophobic, but it has a lot of small amino acids like that in there. And it's amphipathic. So it's possible that this is, and I kind of think it probably is, but none of these viruses has been worked on, so we don't know for any of them. The other thing it could be is this looks vaguely like blue bars, so that might be the other amphipathic helix that would be the fusion peptide. Hard to say. Now, what's the next thing we should find? Ooh, look at that. And we've got acidic amino acids that are conserved for a change. There's nothing, I wouldn't read anything into that. But we've got a big thing, and we've got some conservation, and I see blue bars. So if we're in blue bar jail right there, probably up to about this point, that is all the gigantic heptad repeat number 1 region. Yeah, and look at it. It's immense. It's massive. It's wonderful. This thing might have 1, 2, 3 little hinge regions where it can origami itself up into a little ball. I have no idea. None of these spikes have been solved because none of them have been worked on. Then you get down into the possibly histidine containing region where you're going to have disulfides that are sort of linking up just to sort of keep everything pinched off to the side so that those heptad repeats will be able to come together. You just got to keep everything else squared away, and we do. And you can see there are some H's kind of conserved, but not always, and other ones that are just popping up here and there. Yeah, there's probably a histidine somewhere for all of these, but it's probably not in a particular place for any of these. Okay, more of this region. There's our last little cysteine. I'm not sure where its friend is. Probably there's one that I should find in a line, but I don't see it off the top. And then you have a shortened version, but a very real version of what looks like heptad repeat number 2. And then my alignment cuts off where the homology just goes absolutely to bananas. That would be where we would expect to find the transmembrane, the cholesterol or lipid binding region, something like the MPER of HIV, and the tail with or without acylation sites. All right, so there you go. That's 2 different kinds of spikes. They would both fold up into something like the spike that you see above. This is the human coronavirus HKU1 spike, and they're showing how it can bind to a molecule. And when they are modeling this based on cryo-EM structures, they notice that when the molecule is present, it actually rotates these little receptor binding domains upward. So that's just a little picture of how this thing can move while it is in the process of doing what it does. There's a group of spikes that have this little pocket or a pocket like this that could be activated in this way, and most spikes don't, so this is specific to one like this. Yeah, not relevant to SARS-CoV-2 directly as far as we're aware. All right, that's your tour of the spike. I hope you understand it better now. And yeah, you can see some of the sort of variation. We tend to talk about the spike as if SARS is the only thing, at least I tend to talk about it like that. But there's a lot of variation out there. Nature's been trying a whole bunch of stuff, and yeah, Mother Nature's come up with some pretty cool recipes that all seem to work. So there you go. Thanks very much.